Okay, my talk will be essentially dealing with structure-function relationships, and you will see the topic is rather large, covers half of biology, if you want to take, but I will pick up few examples, and I'll try to tell you that structure can bring answers and to open questions. I said, in terms of introduction, I will concentrate on structure-function relationships, and my main actor will be the tRNA, and try to answer some questions. And structure-function, I want just to give you few introductory data. In terms of information content, main structure information at the atomic level comes from crystallography. And here you have an example, and with the time behind. And this is a GU base pair. That's the resolution, and you see at that resolution you can unambiguously see everything. See the date, 1999. Few years before, this was the electron density. That's the information. I mean, in both cases, this is experimental information. You see, the model is quite correct at that level, quite correct. But, of course, you lack details. In between, you have the resolution, but also you have the quality of the data collection, detectors, and we are right now living a revolution in quality of detector and beam in everything. So everything is moving. So resolution is only one parameter. What you have to see is the quality of the data. And then, of course, it's easier to have high resolution with good structure crystal of small domains than large complexes like the one we saw yesterday with Marat, ribosome, and the other complexes in which we are now more and more involved because of functional correlations. And then when you have this lability of the complex, then the information becomes more difficult, even if the resolution is there. Because what is important is to look at the quality of the information where the question lies. You know, it's not looking at a place where there is a helical stem very well structured if you are interested in the labile loop. It's half a joke, and that happens very often. So here is the main actor, the tRNA we're talking with the three representations. And what is important is this form, and what is important is that this is this was the first structure, 3D structure of a nucleic acid. And it showed the complexity of the arrangement with the stems part, the double helical stems, the loop, the contact. And this is a very stable molecule in terms of RNA, but it is also flexible. And you will see when it is functionally working for the interactions, the flexibility is important. But for example, at the ribosome level, you heard yesterday the lecture of Marat. It's very important that the distance between the anticodon part and this distance is key. You cannot play with that. This is fixed by the machinery. So about 75 angstroms. So it has to be like that. But you will see in order when you don't need that, don't have that functional constraint, tRNA can move. It has to move to adapt to local conditions. In all my following minutes, I will show you how it can adapt. So there are the rules, and there are the possibilities of the molecules. Only the partner. Just quick 2 slides which are slightly related, I mean, clearly related to the talk of Marat yesterday. He showed you nicely, you know, the important steps, what the high resolution data on the conformation and recognition are very important. He mentioned that distance, what he did extend too much, he just mentioned, he said that, but the tRNA part. I will show you the structure. This is a crystal structure for tRNA. This is the envelope. This is not density. Here is the electron density. with the EFTU on top. Just show the density. And you see when you have the complex with EFTU which is going to the ribosome, it is open, is freely accessible from everywhere. You see the groves. I will come back to this specificity. And here is the slide that I borrowed from Marat, the proofreading ribosome. He told you yesterday that cognate and near cognate are flexible. He was not guessing. This is information built in the data. You see the electron density of the cognate. I mean, I'm sure they have better density now, but this is already quite speaking. Here is the cognate part. You see everything, density, no detail. And just the near cognate, you see, disappears. That shows flexibility, several conformation averaging. It's not well fixed. It's the same site functionally, two different situations. Just by looking at that, you don't need to know anything about function. You know that there is something happening there to the tRNA. And the information goes from here to here. And this has to be, it's highly constrained in terms of positioning. A lifetime. Just I want to show you, where is the information. So this information is not just guess and modeling or interpretation. Now I switch to the core of my presentation. And the part of tRNA also concentrating in the first step of translation of the genetic code. And here you see that is the amino acid tRNA. The tRNA is here. This is the charged tRNA. The charging group is here. With the amino acid is here. And it is charged on the ribose. Terminal CCA. Very trivial possibility. Here you have the anticodon. That's where the message is for the genetic code. And here is the amino acid. That's where the amino acid is attached. So there is a strict correlation between that and that. But in space they are far. We'll see how does that work. You saw yesterday, actually I skip that because that at the ribosome level you have the recognition. Anticodon is fixed in place, and the amino acid is in a fixed place. You have the two sides are fixed, working at the same time. The recognition is operating at the same time together, and the decoding part is really fixed by anticodon recognition. In this place you will see for tRNAs and the synthetases, which are proteins that do translate the genetic code, they make the correlation. You will see that is not really something necessary. But let's go to what happens. It's fixed. As I said before, the amino acid is attached on the 3' and here you have the peculiarity of the RNA helices. The conformation of the ribose here is the key determinant of the flexibility of the molecule, the DNA or RNA. So the C2' endo, which is a conformation which is very, very easily often found with DNA, is something non-standard in non-helical region. It's something very more rare, but it's possible. The flexibility, the importance of the flexibility is shown in this thing. When you go for the C2' endo to the C3' endo, you're flipping the pucker of the ribose, and you see the movement of the bases. They go from there to there, and the ribose. Just by flipping, you have very large variations. So this is a very sensitive place, and by playing with this conformation, and you don't have two conformations, you have a continuum. This is what the figure shows. And then you can adapt. And of course, in the helical stem, here you have the two extreme cases, RNA and bDNA. characterized by the width of the major groove here, and it is well accepted, and everybody thinks that major groove is not accessible in RNA. Well, it is accessible here. It's only the minor groove which is accessible. That's something more or less accepted. But when you have interactions, it happens, and you will see examples, I will show you examples, where the protein, or other actors actually, they go into the major groove. And there, the major groove is opening. So again, another lesson is RNA is not rigid, it's flexible. It can adapt. We'll see how it can. Of course, I can give you the clue first. You know, in a long stem, helical stem, rigidly fixed, it is difficult. It has to have some flexibility. But you will see in the tRNA, when you reach the anticodon loop, for example, or the CC stem, you are at the end of the loop. And there, this is more at the end when you close, it's close to an opening. And then you can propagate. Keep that in mind. These are principles that we illustrate. Now, just another way to change the way of thinking. This is the genetic code, the way it is represented. Actually, the color code refers to something I will speak more at length on Thursday. There are two classes of synthetase that charge the amino acid on the tRNA, class 1 and 2. They have functional differences, we'll see very different. One charges the 3', the other one on the 2'. This is a fundamental difference in terms of chemistry. And so they are a related question. But what is interesting for the purpose of our talk today, you know, if you change, that's a classical representation. If you change the representation, you see the color code relates to class 1 and class 2. You see that you find within the code correlation which were unexpected in terms of evolution. I have no time to go into that, but what is important is that there are 20 amino acids. We have a few exceptions, but okay, we are not talking about, there is always, every rule has exception. 20 amino acids, tRNA synthetase, many more codons. And there are a lot of tRNAs. There are isoacceptors. So the correlation between the amino acid and the codon cannot be rigidly fixed. So the anticodon, what I'm saying in other words, what bears the message is not a key element for discrimination and recognition. Sometimes it is, sometimes it is not. So for the tRNA, the recognition by the synthetase, which is the key step for the translation of the genetic code, what is important is the identity determinant. And these identity determinants are related to the message, but they can be, they have no relationship in space with the anticodon. Here you have an example, the aspartate identity sets. You see in yeast, E. coli, Thermus thermophilus, you have the identity determinants which are here. And in this case, you have identity determinants which are conserved, but here you have a GU, base pair here is a GC. So you see, when you change species, you know, what does identity determinant means? Is that if you change this identity determinant in this, even if you keep that, you will not transfer the message, even with the right anticodon. So these are really identity determinant for the synthetase. So this is a very important concept which was found by one of our chair quite some time ago, one of the speakers, not the chair. Now, how does that work? For example, in the case of aspartate tRNA synthetase, you see the identity determinants are here, 1, 2, 3. That's from yeast. I said, so you have the active site here where the CCA end goes, and the anticodon here. Here we have the anticodon, so we have a strong recognition pattern at the anticodon level. But message is here, the anticodon is there. There is a dynamical conformation. I don't go too much into the details, but there is a communication. In this case, you see the protein enters the major groove site. So there is a dynamic and opening. In E. coli, that's the real complex, the two tRNAs. You see the identity determinant here, the major one and the minor. That is, the minor can be adapted to the loose. The two recognition sites, which are far away, and in E. coli, like in prokaryote, they have an additional domain for recognition of this stem. So for each identity determinant, you have a protein contact and a recognition. But the correlation between that and that is not constant. Here you have an example where you have the monomer. That level you see, when you enter the specificity, you have to find the right interaction which explains that. For that level here you have a GU base pair, here you have a GU, and the recognition of the guanosine, which is the determinant here, is highly specific with this loop. So you understand with the structure that if you disrupt these interactions, you lose the identity. So not only are these elements important, but all the elements are important. It's like a machine that you want to adapt. If you change just one point, you have a slight differences and you lose it. So you need more than one anchoring point to really position it carefully. So this is basic molecular mechanics. Bear that in mind. Usually you have more than one identity determinant, and not necessarily the anticodon where the message, the nucleic acid message is there. But you will see every rule has its exception. Now, the pathway. I told you, you have the tRNA is a dynamic molecule. So this is a summary of several different crystal structures and data that show that the first step for this case is recognition of the anticodon on this part. And then the dynamic goes in that way. The second step, there is a conformational change. That's the advantage of the RNA structure. It's flexible but also rigid. So when you have that, you stack, you bring that here. And this loop, which was open here, closes. And you position here. And once you are positioned, you can, here you have two representations of the two cases. You can run the reaction. Then the reaction is there. Either you are in a case where you have no necessity to check out, no proofreading, and some cases where you have to make the proofreading. So we'll see later how that works. But in this case, we don't need to check because aspartate at the level of the amino acid has no problem. There is no ambiguity. So there is no need for proofreading. So when it reaches that point, usually it's ready to be catched by FTU and go further. So this is more or less a canonical way of the dynamic of tRNA. But just now, I mean, the previous data are already have some year old. These are much more recent data published last year by the group of Richard Gégé and Catherine Florence in Strasbourg. And here you have the human aspartate synthetase with the E. coli, human here, and E. coli on the right. So that is very important also for, aside, I will not concentrate a lot on that, but for medical application, as you all know, it's very good to know the differences between human and other species, either mainly for toxicity or for just activity. Because, so the human enzyme is a good target of structure investigation. So the human here is on the left and the right. What you see here, the right is E. coli. The domain That's a view, one view. You have to compare with this one. That the tRNA is on top, model. And you see that the human has this domain, which I showed before was not present in yeast, but was present in prokaryotes, in E. coli. So sometimes you have to be careful, and it's very good to have the analysis, the sequence analysis, because the rule, when you need an extra domain, at the beginning, we didn't have so many information, so the yeast was supposed to be very similar. It's a eukaryote, very similar to the human, and there was a major difference with prokaryote. And you see here, this is not the case. That's the tRNA in the side, and there is an identity element on that stem. Here's another view. Here you can just rotate, and you see the tRNA on the top entering, and that is the mapping. That's the protein alone, which shows the anchoring point. So that's a charge distribution on the surface. That tells you also another information which is obvious. Nucleic acids are recognized by protein through charge distribution for the first screening. And then, of course, the second screening, identity recognition with hydrogen bond, is more strict. Now let's go a bit further in complexity. You realize that this data, we are dealing with a very old problem at tRNA and structure-function relationships, and data tend to come back and accumulate. Now we have an interesting problem with the tRNA, a very specific tRNA, the selenocysteine tRNA. And this tRNA has one feature, which is a long extra arm. So the canonical tRNA is like that, and usually this arm is rather short. For the selenocysteine, like the serine tRNA, they both have a long extra arm. And here you have the serine, and this is the selenocysteine from different organisms, human, archaea, and bacteria. So the distribution is the same, selenocysteine and serine, with a very long extra arm. Why is that? If you look at the synthetase, there is one synthetase which does the work for everybody, charges all the tRNA. So we have a typical problem. We have lots of tRNAs, we have several anticodons, and we have one protein which has been selected. It does that. So how does the protein achieve that? The best thing, with a canonical tRNA, with a small extra arm, it will need to have several contact interactions. But it is difficult because these points, with the canonical tRNA, there are a lot of points which are similar to others. So the discrimination, the identity, becomes a very difficult target. So to change the structure of the tRNA in such a way that you have two constraints that can be released, that can be solved. One is the identity, and don't forget this tRNA is also going on the ribosome. And this tRNA here, you see the structure is very large. Usually you have that, and here is the extra arm. So this is the clover leaf, and here is the 3D structure. So it's not like that, it's really pointing out. So I don't talk about the problem for the ribosome. It's an interesting question, actually, but for the serine at least. And here is the complex. Let's see. That's another take-home lesson. This is a crystal structure of the complex, but in this structure, the CCA arm does not go in the active site. So it's another take-home lesson. Even if I'm preaching for a very strong, powerful data, seeing is believing. So if you see that, you have two things. two possibilities. You believe it, and you say, well, there is another protein coming to charge. Or you say, no, I have a packing artifact. So that's another question. When you have a structure, is true for all methods. You need controls. So the interaction here with this extra arm by this domain is happily correct, because we have the structure with a serine complex and tRNA is here, and that one is correct. That is correct. It is a recognition and the stem. In this case, we don't have. If you recognize here, you don't have. So that shows you, even from artifact, you can get informations. That there is a flexibility, but the time life of the active form may not be too strong in absence of other cofactor. And then if you have competing factor, the packing, you can release that. So you have a dynamic aspect. So it's always like that. And thermodynamics and dynamics are key players. You are recognizing, and we come back to that our example, you have first to recognize to get the right position and to get everything ready. But then you have to stabilize the complex. When I say you, when nature has to stabilize in order to give the complex time for the reaction, and the thermodynamic has to be there. Look at the tertiary structures of the tRNA, and you see here the different, that's tRNA sec from different species. And you see the principle. This is the tRNA. That's the skeleton, that's the surface. That gives you a better appreciation of the difference in structure. They are the same things, but you see the differences here are quite significant. So which means that what is important is what is conserved, because they are all charged by similar synthetase. Of course not the same, because organisms are different, but there are small adaptations. So the recognition is there and not here. So the recognition is here. So you have one anchoring point here and the other here. So now I come to one last, one example important. This is the alanine-tRNA-Synthetase from archaeon. The data I have presenting you are from Yokoyama laboratory. Just gave that me some time ago. I just now think I can use because the data, the results are in press now. tRNA-Synthetase was an exception. It's a class 2 synthetase. I will come back to that in France. In terms of class 2 synthetases, there are common rules. The active site has to be the same. So that's the key separation. And then the recognition of the tRNA is similar. Class 2 synthetases are dimers, obligate dimers. And there were two exceptions to that rule. Yes, and they are charging the tRNA on the 3' hydroxyl group of the terminal CCA, which is a characteristic. So they have common. They are about 10, 11 actually, because there is an extra amino acids in that class. And all these synthetases, obligate dimers, which are on 3', there were two exceptions. There is still one. Alanine-tRNA-Synthetase is a monomer, or a dimer, and in some cases even a tetramer. So basically, the dimeric structure conserved in all synthetases is not conserved. This is a very important point. So mechanistically, I think has some implication. But more importantly, for the point of view of my talk today, this synthetase, a specific in term, I told you about the identity determinant. The specificity of that system is that the the tRNA identity determinant is one single base pair in the acceptor stem, the GU in the third place. So I told you, you do the rules. In order to fix an object correctly, to anchor it, you need more than one point, which are trivial observation, and it's true for all the others. And this may not be the anticodon, but it has to fix in order to position correctly. And so this was a puzzling. It was so much of a puzzling that actually when the results were published, they were giving speculation to a second genetic code, if I remember. So RNA base. What did I say? This field is full of main discovery that don't last forever. Now that the structure is important, the structure brings the explanation. Why can it work? So there are two aspects. First, the tRNA is here. So in archaeon, it's an archaeobacterium. It brings the answer to one question. First, you see the binding here. Oh, that's the editing site for coming back to. There is a dynamic here. I got this movie from Yuki Yokoyama. You see the oscillation here is there. That's the anchoring. Goes from after that to the editing, because there is an editing necessity for proofreading. So it moves, the tRNA moves. This aspect, you see, the structure, if it depicts the reality, needs to have dynamic behavior, so to transfer the tRNA, which is also, again, a difference with all the other synthetases. These other synthetases, I will give you an example later. It's a more, let's say, thermodynamically safe. The other point is that the dimer here, you see the two active sites are far away. In all synthetases, they are connected. For this part, there is no explanation yet. Why is that? So that is not. But what is important is the recognition. So I will concentrate on this form, amino acylation editing. So you see it flips from one to another. But this amino acylation part, which is very important, because then you have to recognize the tRNA and to do the reaction. Just to concentrate a little bit, you see a comparison between the aspartic acid and the alanine. Why these two? Because they are two class 2. Yeah, most of the rules on the class 2 were based on the aspartic observation. You see, by keeping the active site domain in the same position, we superpose the active site, and you see the tRNA here, aspartic tRNA is here. Of course, they are, the rest of the protein is there, and the tRNA alanine. They are significantly different, although the end is the same. And to show that this is general, you see when you superpose all tRNAs of class 2 with respect to the active site is like that. The tRNA ala is moved. So there is this sign. So that could be general, but it's an exception. And I tell you, here we are dealing with exception to a very fundamental rule. But the more important is the fact that I told you that here there is one base pair, which is the recognition element. How can one base pair work? And this is the answer. You see, the anchoring point is the GU base pair. For many years, there are lots of papers in the field column. It was well accepted, and actually I started with that. That is the GU. If you look at the GU in a stem, what is really specific in terms of recognition is the minor groove. Long speculation, you go back to these. Yeah, well, other people, MacLean and other people. There are a lot of Smartfish, Schimmel, MacLean have been working, many Ulysses, lots of papers in the most prestigious journals to explain mutation. This is the anchoring. Everybody was hammering. When the structure, we solved the structure of the threonyl-tRNA, I show you later, another example of editing, we had this, the recognition domain, which is similar to, in terms of sequence, coming on the minor side. So everything seemed to fit, but we still had a problem, one base pair. And if you change the base pair, you lose the identity. There is one thing that maybe people didn't notice too much, at least I didn't. Well, maybe not the only one. That the binding constants were conserved. So the mutation didn't alter the change most of the KD. So you could change the mutation and still bind. So it was not a thermodynamic stability complex. So the complex was not changed. So that should have, that argues again the minor groove binding and recognition. So you have a molecule where if you mutate, you can still bind, but you don't charge. It's a very interesting puzzle. What happens? The solution is there. And seeing is believing, but this time we have data. The recognition is through the major groove. The binding, sorry. And the binding is very strong. There is also a minor groove recognition, but not so important. Which means that if you mutate, you don't change interaction, the contact. The specificity, of course, major groove is not so important, which was the address the puzzling answer. So you bind, you anchor, you're still fixed. The other you can change. So you are anchoring the same machinery in terms of mechanics, and one works, the other not. You're anchoring them at the same point. And now we are back to something which is, after all, trivial for people in the RNA world. Should be. But nobody had that solution. Once you have that, you anchor the stem. You fix everything. What's different? In one case you have a GU, in another case you have AU, GC, or whatever. What's on crick? What is specific of nucleic acids? On the CCA end, you have some freedom here at the end. The stacking is changing. So the basis, this is a pure RNA situation. You have slight changing. In order to disrupt a very fine-tuned mechanism, you don't need to change everything. You need to go to the active site. If you are slightly off, you are off. That's true even at the ribosome level, isn't it? Close to the truth is not the truth, or a truth situation. So the mechanism, after all, is simple, is a K-cut mechanism. That explains why the binding is the same. The stacking being different, and the stacking is sequence specific. GU, UG is not the same, I mean everything. Then you go back to basic nucleic acid structure. Then in one case, you go with the natural base, you go directly to the active site. Here, if you change the stacking free, you bump there and you are out. But the complex is the same, except for these last n. Again, I come back to what I said before. You say, well, you told me at the beginning that major groove is not accessible. That's true. But not at the end of a stem. The end of a stem is flexible. I gave you an example. I didn't expand too much, but we have lots of structures on the aspartic system where the anticodon is recognition. The protein enters the major groove. It opens. It's an entry. At the CCA end, it's flexible. So it can open, can be reached. Of course, if we had a longer stem, then that would be more difficult, or in the middle of the stem would be more difficult. So mechanically it makes sense. It's really RNA structure dynamic. I give you the basic principle, but for me it's like what I said yesterday when the solution was given for the proofreading in the ribosome. When the solution is there, it's very simple, and you have to accept it. I mean, it's just that's life. If you have a complicated example, just to show you the other specific behavior of the tRNA, that's a more classical. I told you about the threonyl-tRNA synthetase. Why is that important? It is interesting because the CCA goes here, and this is the editing domain, the N-terminal domain. This N-terminal domain has a contact zone which is exactly conserved in terms of sequence in ALA tRNA. It's the same. When we found the structure, we realized that. And this is the one coming here in contact. So in the case of threonyl, which is a standard case of editing, you have recognition, minor groove recognition, and then you have a flipping of the amino acid, the CCA charge from here to here, while keeping here the contact area. This domain is found in alanine-tRNA synthesis. Why I said, you know, we have the answer for the mechanistic work of control of the CCA. I'm still puzzled with this dynamic, so it's still open. But this case of recognition and then flipping from one side to another, and that's the tRNA conformation in the glutamine structure, class 1. So just superpose, just to give you the movement, this is a natural movement for tRNA. And if you look at the structure of the proofreading mechanism for the class 1, it's exactly the opposite. You have the active site here and the editing site there. So you have like a mirror. So again, it works for everybody but alanyl so far. In alanyl, the editing domain is somewhere here. That's why you have to do the movement that I showed before. So there is still room for improving our knowledge. Just last thing I will skip, the mitochondrial tRNA, which is rather important for drug design. And it shows that the threonyl here synthetase has a recognition which is not depending on the anticodon, which is, the domain is same, but it recognizes different size of loops. So that's a very puzzling thing. I have no time to go into it. It's interesting how the tRNA, the synthetase can adapt and select the same tRNA, different isoacceptor in mitochondrial. This is just to conclude that the tRNA story, I started right from the beginning. The two classes were already 15 years ago. Well, no, more than that, 25 years, sorry. So old discovery. There are general rule in terms of original genetic code. Most people think that class 2 are the original one. There are reasons, we can discuss that next Thursday. But you see that the rules and the answer are really matching RNAs, a very crucial step of genetic information. And then control, you have charging, proofreading. And this is, although we had the first structure of a tRNA in 1974, first structure of a complex 1990, and 2014 we have an explanation of one of the exceptions. So the field is still moving. We don't have, at least I don't have an explanation for the solution why is that related really to the evolution of the genetic code or even its origin. These are pending fundamental questions, and but we move. It's still up to date, and with a small RNA molecule, by the way, which was used as a template to build more complex, maybe 20 years ago. Now we can we have tools to understand the mechanism of life. Thank you.
Thank you.